back. Move on to our last talk of this session, given by Dr. Fei Wang, Cellular Developmental Biology, University of Illinois. And he will talk to us about the molecular mechanisms governing fate decisions in human embryonic stem cells.
Thank you very much. Well, first of all, thanks for coming. And I'd like to thank Jimmy and Irfan for giving me this opportunity to present the work currently going on in my lab. As you can tell here, the major goal of my lab is to understand the molecular mechanism underlying the fate decision of human ESLs. Well, you may wonder why I'm here, right? And how does the research project in my lab fit into the overall theme of this workshop, right, which is biosensing and bioactuation. So, well, I can give you a couple of reasons. Number one, this pluripotent stem cell field, which includes human ESLs. I'll use human ESLs for the rest of my talk, and you know this, it stands for human embryonic stem cells, as well as human iPSLs, induced pluripotent stem cells. They together are named or defined as human pluripotent stem cells, have become a very exciting field of research, not only for cell biology, very important, I'm a cell biologist, obviously, but also for tissue engineering. That's the reason why I'm here, because I think there's a great opportunity to bridge the gap between cell biologists as well as engineers. And that's the reason I think Jimmy was actually inviting me in the first place. And the second reason, which is I think as important, is that I've been very fortunate to be part of this the center grant we have at the University of Illinois, together with MIT and Georgia Tech, and this is something we call, EBIS, Emergent Behavior of Integrated Cellular System. So the overall goal of this center grant, STC, is to basically, use cells and put them together to design machines to perform certain purposes. So I'm part of this, very fortunately, part of the center grant. My goal is to basically, making cells or generate cells for these cellular machines. And for that, I'm very grateful for the leadership of, Jimmy and Rashid and Tahir and also Martha, who I believe gave a talk a few days ago. Now, going back to my talk. So, like I said before, the goal of our research is to understand, how these amazing human stem cells maintain their fate decisions or make certain decisions. So what I'm going to do today is to first give you roughly 10 minutes of background. I think this is going to be important for you to appreciate, the general, idea of these cells, and then I'll go to my research project. It's very important that I may give you some detailed information, but it's more important that you get the bigger picture. I think if I can deliver certain bottom line, I'll be very happy. Right. So as many of you probably know already, human ESLs were derived from the blastocyst stage of human embryo. And what you're looking at is the stage of blastocyst. And then there's a group of cells right here in a structure called inner cell mass, or ICM if you read books or papers. So the idea is that people for many years were trying to take these cells away from the inner cell mass and put them on culture dish. I mean, why do you do that? And the reason is because people are hoping that if we do that, we have a sort of a cell line that have all the development potential, as in this particular cell stage, right? They can become differentiated endoderm, mesoderm, or ectoderm, right? All the cells within our body. And then this was very easy on paper, right? I mean, you take the cells and put them on dish, but it took people at least 15 years to figure it out, even after this was done in mouse embryos in the first place. So the first person who did that was someone called Jamie Thompson at the University of Wisconsin-Madison. So he actually graduated here from UofI, from the biophysics program here in 1982, I believe. So he was a product of our UofI education system. So he did this in 1998 and published a landmark science paper. So he was able to take cells from inner cell mass and grow them on dish and establish stable cell line that can grow basically forever. And these cells are called human embryonic stem cells or human ESLs. So why are these cells so special? And why people are excited about these cells? And then I'll give you sort of a rough idea, and so that you can appreciate why this become a very interesting model for cell biologists like myself, right? So first of all, these cells can undergo something called long-term undifferentiated growth. That is, if you grow these cells on a dish, you can basically grow them for at least hundreds of passages. I don't know how many of you grow cells before, right? You grow them, and they become confluent. You tritonize the cells, and you put them in you subculture them, right? So this is amazing because for typical somatic cells, that is, if you isolate cells from human body, they will basically stop growing within 50 passages, actually even less in reality. So, but these cells don't have that problem. They can grow for at least a few hundred passages. So this is why we use the term here long term, because you can grow them basically forever. And then during this sort of undifferentiated growth, they maintain the ability to differentiate into any germ layers in the cell body, the 3 germ layers, like I briefly mentioned before, mesoderm, endoderm, and ectoderm. So they can grow forever, but they can they have the ability to differentiate when you want them to differentiate. Right? So this is called pluripotency, meaning that they have the differentiation potential to become any type of differentiated cells in the body. Now, like I said before, under certain conditions, these cells can differentiate into cells within all these germ layers, 3 germ layers, as well as triple ectoderm. So all you have to remember is this rough idea. These cells have the ability to differentiate into basically any type of the cells in the body, and this is called pluripotency. This is the reason why people are so excited, and then the applications are huge. Number one, this really provides us an interesting model for understanding early human development. And the reason is very simple, right? You cannot do experiment with human, like human, right? You cannot do that during development. So this becomes a very good replacement for doing that. And for instance, you can use these cells to understand birth defects and infertility, and also control of early control of cellular plasticity. Like I said, these cells can adopt different cell fate during the process. They can grow, they can differentiate, and meanwhile they have to maintain undifferentiation while they're growing. Right? So they become also a very good model for understanding cell division. And this is, I think I alluded before briefly, that somatic cells, you can only grow them for 50 or so passages or even less. And if you force them to become immortalized, and then that's bad, and they become eventually malignant or even cancerous, right? But these cells do not have this problem. They can grow, they have very tight mechanism to control their growth so that they won't become cancerous. So how does this work, right? If you can really figure out the growth regulation in these cells, you can really understand how this can become bad in cancer cells, right? So this is, I think, highly underappreciated. I mean, everybody understands cell-based therapy, right? I think we all know that, right? You take these cells, you differentiate into beta cells or neurons, and then you can use them for their therapy. But I think they're also outstanding model for study or understand cell, basic cell biological questions. And then meanwhile, these models can also serve as a shortcut for drug discovery and testing. So what this means is that you can potentially use these cells and differentiate them, for instance, into cardiomyocytes. And then you can, if you have a drug, you can test the toxicity and other, functions using these sort of differentiated cells without sacrificing a lot of mice. I mean, so that's what people are saying. Furthermore, these are human cells. They're not, mouse, right? A human and a mouse are different after all, thankfully. So, so these are really the promises that these cells sort of possess. Any questions so far? Is this all clear? Okay, so but before we really realize all these potentials, there are many hurdles. I only list a few here. Number one, I think even after like 12 years, after these cells were sort of derived, we still have very limited understanding of the condition and factors for long-term cell-free yield. And you say, well, why is that important? Well, the reason it's important is because if you were to use these cells for therapy, you're going to have to generate a large amount, right? And the culture conditions are very expensive. I'll get to that in a minute. So if you're able to understand this question much better, and then you can probably find a cheaper way to do that. I mean, when you talk about cell-based therapy, you're talking about not just your little test tube, that little chunk of cells. You're talking about organ-sized cells, and that really takes a lot, right? So obviously, the conditions for and the factors for efficient direct differentiation are also very understudied. And I use the word here, efficient. People know how to differentiate human ES cells into cardiomyocytes and neurons, but the efficiency is always very low, unfortunately, and then you only see a very small number of cells, and that's bad. And going back to the point I was trying to make, and you need a large amount of cells for therapy, period. All right, so, and the other question, the third question is derivation, characterization, and application of induced pluripotent stem cells. I'm not going to talk about that today, although I think we're actually getting to that as well. But these a couple of questions are the focus of my lab for the time being. Now, before I get to the experimental data, I wanted to share with you some basic culture conditions for growing human ES cells. I think this might be useful if you become interested in this particular field in the future. So the first methods was basically developed by Jamie Thompson, like I said before, in 1998. So the idea is the following. So you take these cells and you grow them on top of feeder cells, which are typically mouse embryonic fibroblasts. So these cells are known as feeder cells. Number one, they provide physical support for embryonic stem cells. And number two, they also make some factors. They secrete some factors that are essential for maintaining the long-term self-renewability of human ES cells. Well, as you probably can understand, this culture condition is very complicated. It involves multiple culture systems. So 3 years later, this company called Geron in California, which also is a major player in human stem cells, basically developed a condition called feeder-free culture condition for human ES cells. So the idea is that they decided to take away the feeder cells, and then they provide a matrix gel, a sort of mixture of extracellular matrix, and put them on the bottom of the dish. So the matrix gel will provide the support. And meanwhile, they use conditioned medium, not meth, but meth conditioned medium, and then combine with other factors to grow these cells. So basically, you have only one type of cells in the system. You still have meth, but not meth themselves, but meth conditioned medium. Conditioned medium meaning that it is the medium where meth are cultured, so they still contain the goodies that are important for culturing human ES cells. Now, very recently, 3 or 4 years ago, Jamie Thompson's group again developed something called feeder-independent culture. So this is called teaser medium, commercially available. This is an entirely chemically defined medium. So what I meant by that is that if you look at meth conditioned medium, you really don't know exactly the components are, right? So they're not chemically defined. You sort of know they're important, but you don't know what exactly are the crucial ingredients in the conditioned medium. But here you have a sort of a chemically defined, and you can even look up the papers to see what's in there. It's actually very tricky to make this medium, and then and we decided, well, initially we wanted to make this ourselves, and it turned out to be quite difficult. So they're commercially available, quite expensive, but very, very powerful. Now, so the particular question at least when I started my lab 4 years ago, was the basically how pluripotency it was regulated in these cells. How are these cells able to maintain differentiation while they're growing? So when I started out, the field is actually very limited. As you can see here, there are 3 major players called Accord, SOX2, and NADOC, and I think this have probably become household names recently. And these are transcription factors that are crucial for your ESL identity. If you take any of these factors away, cells will differentiate. They won't behave as ES cells. But people also know that there's some extracellular factors. So what you're looking at is basically a cell, right? This is just sort of my version of the cell. This is nucleus. You have transcription factors. You have factors outside the cells that are also important. For instance, basic FGF, which is a crucial factor that maintains pluripotency of human ES cells, a crucial ingredient in the culture medium. And then meth conditioned medium, at least in certain cases, and serum replacement. This is sort of a replacement, by definition, replacement for serum in this condition is chemically defined too. So TGF-beta has also been shown to be important in some cases. But what is really lacking here is the intracellular pathway that sort of mediate this, outside-inside signals remain largely unknown. So what are the gap you're seeing here? How does that work? Well, you may say, why? Well, I mean, that's interesting, but why is that important? I hopefully can show you that this is actually very important, even for application of ES cells. So our idea is the following. So we wanted to tease out the major components of the signaling pathway. And for instance, if you're able to find a crucial molecule essential for pluripotency, you can hopefully expand that knowledge to eventually achieve a signaling network. And of course, this is highly exaggerated, as you can tell, but basically we wanted to achieve a much higher level of understanding by teasing out the key component, starting with the key component in the signaling network. And then I'll hopefully make the point, I think this is one of the bottom line I wanted to deliver by my talk, is by understanding how this network works, you can really control the fate of human ESLs. I think this is where you can connect your knowledge, basic science knowledge, to application of these cells for therapy. So we'll get to that, and hopefully you sort of have an idea here. Right? So this is very important. Now, the overall strategy is the following. So it's very straightforward. I'm sure you understand this. So what you can do is the following. So you can grow human ESLs on sort of a pluripotent condition or normal condition, right? With a basic FGF, and CM stands for condition medium from math. So these cells would maintain undifferentiated. They keep growing, they're happy, and they're in green color just to show that they're undifferentiated. And then you can put your treatment here, right? So you can treat the cells with, for instance, inhibitors, pharmacological inhibitors, small molecules, chemical compounds, and also shRNA, or siRNA. So the idea is that by doing this sort of a screening, you're able to identify some key component. And then so that you can, with this sort of a particular treatment, cells become differentiated. So what that means is that whatever you're taking away or you're blocking is very important, right? The cells without it, cell would differentiate. So this is the target of this particular chemical compound or shRNA are potentially positive regulator of pluripotency. Does that make sense? So this is, you take it away, it becomes differentiated, right? So you can do the reverse screen. That is, you grow these cells on matrix gel without basic FGF and condition medium so that the cells will differentiate, right? And they're not very happy to differentiate. And you can put the same treatment here, and you're looking for the one that could reverse this process, right? So basically, whatever the target of this compound is a negative regulator. So if you take away the negative regulator, cells would show delayed differentiation. So by doing this sort of a forward and reverse screen, we're hoping, or at least we were hoping a few years ago, to identify some key components that regulate pluripotency of human ESLs. Right? So this is the general strategy. So we start out by screening a 40-ish, roughly 40 known pharmacological inhibitors. And a lot of people ask me, why did you start with this? Right? I mean, the reason we start with this is we didn't really know what to expect, to be realistic, to be sort of honest. And number two, these pathways, these inhibitors, this in here, and you don't really have to know the detail. They pretty much have known targets in the cell. So whenever we see a fact, we kind of know what their targets are, so make our life a little bit easier. And you will see that when I present my talk all along. So by screening this 40 also inhibitors, we're able to identify 2 positive and 1 negative regulators of human pluripotency in the past 3 or 4 years. So the first target we identify is actually mTOR, mammalian target of rapamycin. The compound that target mTOR is rapamycin, obviously. So this ended up a paper that came out a year ago, essentially. So I will talk about that in my talk for probably 10, 15 minutes or so. And then we also identified blocstatin, which is a very specific inhibitor against non-muscle myosin 2. And you may wonder why that is. Unfortunately, I'm not going to talk about that today. It's a manuscript being reviewed. And then I'll also talk about the third chemical. It's called compound C, or dorsomorphin, by other people. This is actually it's a very good story for us. I will be very happy to share with you how we identify all these targets. So the target, the potential target before was actually AMP activated kinase, so they call AMPK. And it turned out that the target is not AMPK, at least in human ESL. So that became a very interesting story for us. Anyway, so I'll basically present you this story and also the compound C story to make the point that, number one, you can use this approach to understand how these pathways work. And number two, you can use the information to manipulate cell fate for your own benefit. Now, so a little background on mTOR. So this is the mammalian target of rapamycin, very important molecule. That has been heavily studied in the past 10, 20 years. So it's very essential gene. I'm not going to go through the list in great detail. It's a very important signaling molecule. However, the precise mechanism that regulate pluripotency in human ES cells remain unstudied when we set out. So And then the reason, the way we identified, we realized that mTOR is important was basically by this very simple experiment, as I showed you before in our strategy. So you grow human EA cells on a dish, and then you put your 40-ish inhibitors in the cells. And you quickly realize there's something very special about this inhibitor, or rapamycin, which is the inhibitor of mTOR. I'll repeat that. So what you realize that normally the cells will grow as sort of a very compact colony with very well-defined edge. And then, however, just within 48 hours, the morphology begin to change. And then they flatten out, and then they sort of spread out. And then if you study human EA cells, you realize that these cells are differentiating. I mean, this is a very, very strong sign. And then as time goes along, and you see even more stronger differentiation, right? And then what you see morphologically, the cells look like they're differentiating. But how do you show this at a molecular level? What you can do is to do the look at ARC4 and non-organ SOX2, all these famous transcription factors. And this is immunofluorescence. I think Scott probably give you some general idea. So you use antibody to determine the level of these proteins, and you quickly find out that the level is dramatically reduced. And this is initially in the nucleus, as you can see here in this sort of a spotty pattern. And then in many cells, this fluorescence begin to disappear eventually. Of course, you want to do this in a sort of a more quantitative fashion, and this is what we did. And then you look at the level of ARC4 and SOX2 by using Western blot. And then as you can see here, on day 4, the level of ARC4 and SOX2 begin to down-regulate pretty dramatically when compared to control cells. And on day 6, the reduction was even more dramatic. Does that make sense? Okay, and then you can also look at the mRNA level using real time. And the bottom line here is that all these black bars represent rapamycin-treated cells, and they're greatly decreased, indicating that indeed pluripotency genes are reduced, cells are differentiating. Okay, but I want you to call your attention to day 2. So we did a time course analysis, and this turned out to be quite important for future studies. Well, we realized that the time frame that these pluripotency genes begin to downregulate is roughly on day 2. So for the first 24 or 36 hours, we really didn't detect any changes. It was not until roughly 48 hours these genes begin to downregulate. So I'll come back to this point a little bit later on. Okay. Now, so what we have shown so far is the following. So we shown that mTOR is quite important for stabilizing the circuitry of pluripotency. So these 3 transcription factors. And if you take mTOR away using inhibitor, or use knockdown, and we actually knock it down as well, you destabilize the circuitry, and cell begin to differentiate. Right? Very simple. Now, the question becomes, what happens to other aspects of cell fate? Right? As I told you before, when you talk about the fate of the human EA cells, you talk about growth, differentiation, right? We actually show that, and also pluripotency. So we have examined the effect of rapamycin on pluripotency, but there are also other aspects of cell fate we didn't look at before. So we also look at cell growth, right? This is when you talk about long-term self-renew, cell growth is a very important hallmark of growth of long-term self-renew. Right? Well, and also differentiation. If we look at differentiation first, I'll show you the long-term the growth in the next slides. So we know that rapamycin disrupts pluripotency, but what happened to these cells exactly? Do they show direct differentiation or not? So to address the question, we did the following experiment. We put the cells on model layer at rapamycin, and the cells differentiating as I show here in this graphic presentation. And then we look at markers for different germ layers, mesoderm, endoderm, and also ectoderm. And what happens is the following. When you treat the cells with rapamycin, cells begin to differentiate, mostly along mesoderm and ectoderm lineage, endoderm lineage. Do you see that? So the marker go up, indicating that when you take mTOR away, you have more differentiation activity along mesoderm and endoderm, right? So what does that mean? That means that what mTOR normally does is to suppress this differentiation activity. When you take it away, this inhibition is no longer there, it's self-differentiating. Right? So basically, mTOR normally suppresses mesoderm and endoderm activities in human EA cells. Well, and this is also shown in a different differentiation assay, and this is actually very commonly used assay called embryonic body differentiation assay. So this is just for the sake of some knowledge for you. And then when you look at differentiation, you use this we call EB assay. And then what you do is you grow these cells on low adhesion condition, so that they begin to become aggregated in suspension. And then this suspended cells contain cells of all lineages, essentially. So it's a mixture of mesoderm, endoderm, ectoderm cells. And people use this EB as a bar commonly to isolate differentiated cells. And as you can tell, it's not very efficient because it contains differentiated cells from all lineages, right? You have to follow by cell sorting and all these things. But anyway, you can examine the differentiation potential of these cells by using this type of assay. Again, to make a long story short, we find the same thing. That is, mTOR suppresses mesoderm and endoderm activities in a different differentiation assay. Now, going back to growth, we also look at the proliferation of these cells with or without rapamycin. Right? It's very simple assay. You put the cells on dish, you add rapamycin, you look at cell number on different days. So what's shown here is that the effect of rapamycin on growth is very rapid. This is your normal control cells that keep growing essentially, right? And more cells, more time, right? And then if you put rapamycin there, there's a huge reduction in cell growth, and the cell number basically reduced by half. And then you know that this is probably not due to differentiation, because many people will argue, well, the reason you see a reduction in cell numbers is because they differentiate. When they differentiate, they don't grow, so you have a reduction. It's very unlikely to be the case, because when we use retinoic acid, which is known to be able to induce cell differentiation, give a different pattern. So for the first 2 days, cells are going just fine, but then they show a decreased growth, most likely due to the fact that cells are differentiating. So we didn't see that. The inhibition on growth is very rapid. So we infer from the results that mTOR is also important for proliferation. When you take it away, cells cannot grow faster, right? Okay. And you may also argue that, well, there's one possibility. The reason that mTOR is important for differentiation is because they block cell growth in the very beginning, right? So when they slow down the growth, then maybe they will differentiate. So is that a possibility or not? We kind of ruled out that possibility. We use a cell cycle inhibitor called rescobutin here. So this is a cell cycle inhibitor that is known to reduce cell growth. So the idea is that what happens if we just reduce the cell growth per se, right? Then what happens to pluripotency? The answer is that if you just reduce growth by blocking cell cycle, the pluripotency genes remain largely unaltered. It's only when you treat the cells with rapamycin you see reduction in pluripotency. So what does this tell you? And it's sort of a lot of very simple calculation. So our inference from this study is that mTOR is important for 2 aspects of long-term cell growth. Number one, mTOR is important for proliferation. When you take it away, cells don't grow as rapidly as they should. And also it's important for anti-differentiation, for suppressing mesoendoderm activities. And these are 2 separate responses. That really is the inference of the study, because they really have nothing to do with each other, right? So mTOR is important for both. But then the question becomes, well, this is nice, but how do you know how mTOR does this? How does mTOR regulate proliferation and anti-differentiation, right? That became the very obvious question for everyone. Okay, so before we, so we spent some time trying to understand the signaling mechanism by which mTOR regulates pluripotency. So the first thing we did is to basically make a connection between the extracellular factor or extrinsic factor to mTOR. So the question is, does any of these factors regulate mTOR activity or not? Right? So we wanted to, like I said in the very beginning, we wanted to show that mTOR is a very important, intermediate in the cells, right? So how do you show that? You show that mTOR is important for relaying signal from outside cells, and then also relay that signal to the nucleus, and it's important, right? So we basically use a readout for mTOR activity, which is a kinase called S6K1. So it's a well-known downstream kinase of mTOR. So we use that as sort of a readout for mTOR activity. So what we're able to show, again, make a long story short, that any of these extracellular cues are able to activate mTOR, which include, for instance, basic FGF, and also knockout serial replacement. I only showed KSR and also initial medium before. So all these components that are important for maintaining pluripotency are capable of activating mTOR, indicating that mTOR is a very important intermediate in the cells. Does that make sense? Okay, so we're able to make that connection. Now, so we know there's a connection here, but the bigger question that remained was how mTOR were able to regulate differentiation and pluripotency. All right. before I get to that, I'll briefly show you the other strategy which show that mTOR is important. So we know that mTOR can regulate relay signal from outside the cells. But what if we cut off these signals and we enhance mTOR activity? And is this manipulation sufficient to suppress differentiation and protect pluripotency? This is cell signaling 101, basically. If you study cell signaling, you know this is one of our typical AP stasis. To show this is important, you remove that, you jack up this signal, you see what happens, right? So the way we did that is basically overexpress a molecule called RAP, which is a downstream TTPase that can trigger mTOR activity. So without losing you all altogether, the idea is very simple. So you remove all these extracellular factors. You activate mTOR constitutively by overexpressing something that are known to activate it. And what's going to happen to differentiation and pluripotency? And we show that indeed this overexpression of RAP can activate mTOR signaling by this sort of a twofold increase in S6K1, the readout we used before. And then we look at all these differentiation markers as shown before. So this is sort of real time, very complicated, but the bottom line is right here. So if you constitutively activate mTOR in the absence of basic FGF intermediate, this constitutive activation can prevent mesoderm and endoderm activities induced by basic FGF and condition medium withdrawal, consistent with our idea that mTOR normally does is to suppress these two lineages. Right? However, we didn't see a sort of rescue of downregulation of pluripotency marker. There are two possibilities. Number one, there's only twofold increase in mTOR activity. Maybe that's not sufficient. And number two, remember, mTOR doesn't regulate ectoderm, for instance. So the cells are still able to differentiate along that lineages. So the cells can still differentiate and disrupt this pluripotency. So there's some possibilities. But again, the big picture here is that you can do this all kinds of manipulation to dissect the role of a certain molecule in regulating pluripotency. Now, the last part of this particular story is the downstream signals, right? What happened? So we actually had no idea, when we started out. We tried some obvious, molecules that didn't work out. So we had no choice but to basically apply to functional genomics. And we performed genome-wide microarray analysis to dissect what the potential downstream molecules mTOR are in these cells, right? It's a very simple idea. How do you pick the time point? This turned out to be quite important. And going back to the point I made a few slides before, we know that the pluripotency markers begin to go down only after 48 hours or so. So in other words, there's a little gap there, from 0 hour to 24 hours or 36 hours. So we argue that this is the time window we should care, because there might be some important molecules in that time window that turned out to be important for all these effects that mTOR is responsible for. So we did a genome microarray analysis using 12 hours, 24 hours, and 36 hours. The reason to pick multiple time points is basically to make sure that we saw something consistently. And then we also use triplicate and to make sure it's statistically important. So this is sort of a cartoon showing the genes we detect. So if you narrow down the parameter, so if you use triplicate and you define changes that more than twofold, you actually don't find too many genes because you do a very early time course, right? 12 hours, which is very early. But what really caught our attention are a few genes shown here. So we found very rapid upregulation of a subset of differentiation genes listed in here. So for instance, T genes, MixL1 and PITX2 and also EBF21. So without, these genes are not important. You absolutely don't have to remember this. But what is really interesting for us is that we realized that these genes are known to be important for mesoendoderm activities. So everything began to come together in our sense. So we realized that the reason why mTOR is important is because they can suppress the expression of these genes, right? So if you take the suppression away and they just go up, and then induce differentiation, and they happen to be mesoendoderm regulatory genes, right? So everything makes sense. But how do you show this is actually causal, right? Microarray is great, and you can show this, upregulation. But does this upregulation, or is this upregulation responsible for the disruption of pluripotency? So we only express one or two genes on that list. One is actually EDF2, and also one is MixL. So the bottom line here is that, yes, if you only express these differentiation genes, it can indeed disrupt the pluripotency. you see downregulation of ARC4, SOC2, and non-ARC cells. So there's actually a causal relationship. It's not just that you saw this upregulation. The regulation can indeed lead to the loss of pluripotency. Right? Okay. What about proliferation? Right? I said there's two arc that mTOR can regulate. One is repress differentiation. The other one is to promote different proliferation. So we also identify two genes. One is cyclin G2. The other one is programmed cell death 4, PDC4, which are known to be growth inhibitory genes. And then it makes sense because mTOR can suppress the expression of these two genes, and when mTOR is not there, and these genes go up, which in turn, suppress proliferation. Right? So how do you show that? Again, you can just use a very simple overexpressing experiment. You can overexpress PDC4 and cyclin G2 in the cells. Again, you can see a very robust reduction in growth. Okay, after we overexpress cyclin G2 and PDC4. So this is really the summary slides. And so by putting all these pieces of evidence together, we came up with this model. Well, we think mTOR is an essential molecule, signaling molecule that regulates pluripotency in human DNA cells. In this scenario presented in the slides, mTOR can relay signals from outside the cells, basic FGF, methylation medium chain replacement, which in turn suppress differentiation, mesoendodermal differentiation genes, and also growth inhibitory genes, for instance, cyclin G2 and PDC4. And this in turn is essential for safeguard the pluripotency of human DNA cells. Okay. I hope this is a sort of simple model for you to understand. But one thing I wanted to make is the following. We mentioned that in the paper, but really some of the reviewers were not very happy. But we think that the pluripotency network is very complicated. And everybody knows that ARC4 and SOC2 are very important. There's no doubt about it. I mean, you take it away, cells differentiate. But I just don't think that this is the only circuitry that is important. And if you look at this sort of scenario, you realize that mTOR can pretty much suppress this subset of differentiation genes separately from ARC4 and non-ARC SOC2, right? As I showed before, if you take this away, this is stable for a little while, but this is begin to go up. So it's not like ARC4, SOC2, and ARC are important for everything. They just basically safeguard all the possibility, all the differentiation genes. They're little pocket or circuitry that can do its own job, right? So, suppressing certain genes. We envision that this little sort of a nodule can work together to suppress differentiation altogether. Well, of course, people don't like the idea that this is not as important as they would believe. But anyway, so this is just sort of an idea we presented before. Now I'm going to move on to the second story. Oh, go ahead.
So you had two other molecules that you
yes,
saw.
This is the second molecule actually.
Right. So do you believe that myosin and mTOR, are these all in the same circuit somewhere affecting each other, or do you think they're
very
separate?
good question, by the way. So why are you here? Anyway, this is one of the students in my department. So, gosh. So, myosin 2 is supported in a separate paper that is under review by Journal of Cell Biology. It turned out that myosin is important for maintaining cell-cell adhesion. So that was the story. So you can intermediate cell adhesion. We really haven't looked at the interplay among all these signaling pathways. I have no doubt that they will converge at certain point. So, and then there's a paper recently showing that even mechanical factors can feedback to TGF-beta pathway. So, I think there'll be a bunch of questions to look in the future. But the first step is to understand how this pathway works to begin with. Right? So, good question. Right. So I'll move on to the second molecule called compound C. As I briefly showed you before, compound C can suppress differentiation, essentially. So this is the original data we did. So what we did is the following. So we grow human DNA cells in the absence of basic FGF and conditioned medium for 6 days. And what happens that, this is just KSR, showing that they don't have basic FGF or conditioned medium. They're just, the only major component that is there is a knockout serum replacement, or KSR. So the cells will differentiate, that's everybody understands that. But when we add compound C to KSR, we see pretty much a large degree of protection of ARC4 and SOC2. So if you look at this particular resin block here, ARC4 and SOC2 will go down after the removal of conditioned medium basic FGF, which is shown before. And then if you add compound C, you see this big rescue, indicating that somehow compound C can prevent the cells from differentiation at least for a little while, right? And then the question becomes, how does compound C do that, right? So again, some basic assays turn out to be very useful for understanding how this works, right? This is a simple differentiation assay I showed you before. The goal of this study was to look at what lineage compound C can suppress. So we know it suppresses differentiation for sure, but we don't know what lineage this compound can suppress. So we use this sort of a monolayer assay and an EB assay to look at differentiation, right? Under EB conditions, differentiation marker will go up very rapidly, self-differentiate, no doubt about it. But if you add compound C, what happens is that the markers for meso endoderms are dramatically reduced. The markers for ectoderm, however, neuroectoderm, is unaltered, actually go up a little bit. So the conclusion is that compound C inhibit mesoderm and endoderm and also tropoectoderm activities. So remember, when cells are able to differentiate, there are basically 4 major lineages they can differentiate into. One is endoderm, mesoderm, tropoectoderm, and neuroectoderm. So 4 lineages. So it appears that this compound can block very strongly these 3. So what does this tell you? This compound can block 3 lineages without affecting the other, or actually slightly promoting the other. What would you do, right? So it appeared to us there's a great opportunity here. Well, maybe we can use this compound to promote neurodifferentiation. That's really the idea, right? So imagine that each cell differentiation is like multiple stream of water, right? You have 4 streams going, in different directions. And if this compound can stop 3, right? What's going to happen to the force? It's going to be promoted, or at least sort of become very strong. Right? So that's the idea. So we follow this idea, and then develop a protocol for neurodifferentiation. And this is the idea. So we culture the cells as single cells at high density, and then grow them to complacency. So this has been shown before that this somehow are this condition is in favor of neuroectoderm differentiation. And then we withdraw the cells. We talk basic FGF and medium from the cells. And then we either add compound C or not add compound C. And then we look at neuroprogenitor markers and see whether this manipulation can induce high efficiency neurodifferentiation. Does that make sense? So this is another example why this study is important. It's not like you just understand basic science. If you know the feature of these compounds, you can really use them together for your own benefit, in this case, to induce neurodifferentiation. So to make the last story short, we used PAC6 as neuroprogenitor markers. We saw nearly homogeneous expression of PAC6 in the cells. We also use many other markers of neuroprogenitor cells. We basically saw the same thing. So the bottom line here is that by using this compound, we can achieve almost homogeneous neuroprogenitor cell formation by using Yang cells. This is the FACS flow cytometry analysis showing that the percentages can reach as high as 90%. Right? So it's very dramatic. I think it's the first compound that have been reported that can basically induce neural differentiation. And then we show that this compound can do the same thing in iPS cells because induced pluripotent cells are the cells that more amenable for therapy. So we show the same thing, that the percentage can reach up to 70 to 80%. So it's a very powerful way to induce neurodifferentiation. We show that these cells, these neuroprogenitor cells, have the potential to become more mature neurons, and this is the idea. So you take these neuroprogenitor cells, NP stands for neuroprogenitor cells, and then you add sonic Hitchcock for a few days. This is called neuropatterning. So you can basically achieve nearly homogeneous, beta-3 tubulin expressing cells, and then you can induce terminal differentiation, and you can see the formation of certain mature neurons, for instance, in this case, EA neurons. And so this is really a nice way to accomplish the first step of differentiation, that is neuroprogenitor. And I think the immediate question for us of my lab is to develop conditions to carry this out to induce high efficiency production of a particular neuron subtypes. Right? So we don't know the answer just yet, but we're able to do this quite convincingly and effectively. Now, what's the target of this compound, right? So you see, well, that it's nice that it does something in these human Yang cells, but what is the molecular target of this compound? So this is where the story became really frustrating in the beginning for us, and also very interesting in the end. So people know for many years that the target of compound C is AMPK, AMP activated kinase. So I took a quote from this company, EMP Chemicals, which is actually a very big company. So compound C is a salt-permeable compound that acts as a potent selective, reversible, and anti-competitive inhibitor of AMPK. And then I'll show you data that there's nothing selective about this compound in a few slides. And then AMPK is a multimeric protein complex that regulates cellular and organismal metabolism, right? And then there's some doubt about this compound. For instance, in this fast letter paper, people show that if you knock out AMPK and then you apply compound C, you still see some effect. So there's something more than just AMPK. And there's a more recent Nature Chemical Biology paper showing that compound C can target, which is type 1 BMP receptor. So the BMP receptor, I'll come back, and it's a family of TGF-beta receptors. Okay, and then they call this same compound as dorsal-morphin, essentially, because it inhibits dorsalization in zebrafish. So we really wanted to achieve a more comprehensive understanding of the target, potential target of these particular compounds. To do this, we basically use this approach developed by Fabian et al., published in Nature Biotechnology. So this is the idea. This is basically a way to identify the potential target of a kinase inhibitor. So what you can do is the following. So you can put some chemical compound that are known to be able to bind to a kinase, right? And then normally you can see this as sort of a culture dish. So you immobilize chemical compound in the dish, and you add kinase. And if this inhibitor blocks the kinase by binding to the ATP pocket, you're going to see this kinase settle down on the dish, right? Because it binds to the chemical. And then you can determine the effect of your chemical compound by just putting it in the reaction. So if there's a competition, you're going to see deattachment of this particular kinase in the suspension. They no longer basically attach to the bottom of the dish, and they deattach. So you can measure the amount of deattached kinases by using a PCR, because they put a DNA tag very smartly to the kinase. So you can determine the specificity or the effectiveness of a particular compound. So imagine you have a compound C here, right? So you basically add compound C to the reaction. And by determining what kinases can be deattached, you basically have an idea of what their targets are in the cells, right? So we actually find more than 60 targets of compound C in the cells. So there's nothing selective, nothing selective about this. I actually believe that for any kinase, in the future, for any claimed kinase inhibitor, something like this has to be done. So basically, they scan the whole kind of 450 also kinases to look for the effect of this compound C, to look for the effect of compound C on all the kinases. So there's 60 potential targets in each one of these, right? So what happened here is that it turned out that TGF-beta receptor families are a major target of this particular compound. And I'll just give you a rough idea. There's multiple members of this TGF-beta receptor family that can be targeted by the compound. Well, we ruled out that AMPK is the target by using RNA-mediated knockdown. So we knock down AMPK, we see something opposite. Not only we didn't see protection of frequency, we see decrease in SOX2, indicating that this is not the target. You can forget about this altogether. And PDGF also turned out to be a major target, and then we knock it down, only seeing a slight decrease in growth. So this is not something we saw with compound C. So that's not the target, at least in human cells. So we begin to focus on the TGF-beta family receptors. And then we know that it has been shown previously that activin pathway and also BMP pathway are involved in the regulation of neurodifferentiation. In that, if you block activin pathway or BMP pathway, you enhance neuroactivities. So that's the reason we begin to focus on these two pathways. So indeed, the bottom line, I'm not going to go through the whole slides. So indeed, compound C can block activin signaling in these cells. Right? So again, I won't go through the slides. There are multiple ways to test that. And the reason we think it's capable of doing that is to basically block these two receptors in this pathway. Now, the compound C can also block BMP pathways in human cells. Again, I won't get into the detail. The reason it's very potent for BMP pathway is because it blocks at least 5 receptors, and no wonder it blocks this pathway very strongly. Now, the other reason why compound C is so potent is because it targets multiple receptors, like I said before, and then we show this is important. So the idea is the following, you probably don't have to look at this data because you probably get confused. So if you knock down one receptor, so remember, BMP signaling pathway, there are multiple receptors. If you knock down only one receptor, you won't see a thing. Nothing happened. There's no inhibition. You have to knock down at least 2 or maybe 3 to see robust inhibition. I think we got lucky here because compound C not only target TGF-beta receptors, it target multiple receptors, and therefore it's so potent on signal. So we show that if you knock down 1, nothing happened. You knock down 2, you saw something. Knocking down 3, you see something very similar to compound C. So this is the reason why compound C is so potent. So it's summarized in the slides essentially. So we think the reason compound C can promote neurodifferentiation is the following. So it's very potent in blocking the TGF-beta superfamily receptors, and then which in turn block endoderm, mesoderm, and triple activin pathways. These 2 pathways altogether can promote these 3 lineages, and then if you take away these 2 pathways, and then you block these 2, this phasization of ES cells, and neuroactivin become basically the only pathway that is still active, right? So and then this idea was confirmed by the combined treatment of SP inhibitor or noggin. So SP inhibitor take away this pathway, and noggin take away this pathway. If you only use one of them, you see only very weak neurodifferentiation. Only when you combine this together, you see a stronger neurodifferentiation, indicating that the reason you see a very strong neurodifferentiation is because this compound happens to be able to block multiple pathways. So it's not specific, but you can benefit from the lack of specificity in this case. Okay, so basically the bottom line here is that this is the first compound that induces high efficiency neuroconversion in pluripotent stem cells. And number 2, we developed a single-step, cost-effective methods that induce neuroconversion under chemically defined medium. So we're really excited and we wanted to continue this biome study supported by IBIX, by this center grant, to find ways to further differentiate these neuroprogenitor cells to mature neurons. So I'll finish my talk. I think I'll skip this. 5 minutes or something. Okay. So it turned out you can also use compound C to identify some major targets to understand basic biology. So the idea of this slide is basically to understand what genes that compound C can block, right? And then the idea is that if you can identify these genes happen to be important in regulating the phasization of ES cells. So to do this, we again apply to functional genomics, microarray analysis. And then to make a long story short, we identified a group of genes that are downregulated with the treatment of compound C, indicating that these genes are normally active, but if you put compound C there, the expression will go down, right? So they're the target genes of compound C. So one of them is actually MX2. So this turned out to be a very important gene. It's called muscle segment homeobox genes. Again, the detail is not important. But by using this approach, we're able to identify MX2 as a key factor of BMP signaling in ES cells. Again, just by using this combination approach, you can really achieve better understanding of how ES cells work. And you can also essentially use this chemical compound for your own benefit. That really is the bottom line I wanted to deliver here. So I have just 3 or 4 slides. I'll just finish up. So as you probably remember, we only, in our initial screening, we only look at 40 or so inhibitors. And clearly this strategy is very, very effective. So we ask ourselves, well, why don't we just expand it to a sort of a screen, to expand it to a library of chemical compounds, right? So we did that actually. So this is, unfortunately for large-scale compound screening, you cannot use ARC4 Western. For, imagine 200,000 compound, you're going to do 200,000 Western blot. That's just not going to work, right? So and there's limited number of pharmacological inhibitors, and traditional readout, Western, etc., doesn't work. And this is well known. I think you guys know this already. So what you can do is to use a fluorescence-based reporter for pluripotency. For instance, you can take this 4 kb promoter of ARC4 and attach it to GMP. So, and it's been shown by us and also by others that this fragment of ARC4 can recap the endogenous ARC4 activity. So when ARC4 activity is going down, you lose GMP. So you can see this in live cells in large scale. So by using this very simple reporter assay, we can screen for chemical compound that could induce the cells to differentiate or induce ARC4 to downregulate. So this is to show that this reporter is working, and then all these chemical compounds that are known to basically downregulate ARC4 works in this assay, in our assay. The reporter is really good, it works really well. And then before we apply for large scale screening, we show that this is feasible too. So if you put the cells in the 96-fold phase, and you can detect the changes just by basically fluorescence readers, and you can see this changes, right? So this is our general strategy. So we're looking for something like this. So this is the real condition. This is just sort of a graph, right? In control cells, you can see a lot of green cells, as you see here. And then for compounds that doesn't exert any effect, you can see something very similar between these two. And in many, many cases, compound can induce cell death. So all the cells are basically disappear. You don't see GIP, but they're false positive, right? They kill the cells, of course you don't see GIP. And then there are also cell growth inhibitors, right? I mean, you see a reduction in GIP, you're happy, but wait a second. They're able to reduce growth, therefore you see a smaller number of green cells. And this is something you're looking for, right? You see cells still there, and which is sort of hard to see just looking at fluorescence. But they're no longer fluorescent. That's the hits we're looking for. And this happens to be only, it has only probably 0.1%. So one compound out of 1,000 compounds can do this. So this is a very brief work by in my lab, who screened basically 200,000 compounds available in a UIUC high throughput screening facility. Amazing guy. I mean, I would not do this. I really give a lot of credit to my students. So this is he spent probably 1 year actually screening all the compounds in the library. He essentially find 120 or so chemical compounds that could induce MARC4 to downregulate, not only in the reporter level but also in the endogenous level. So he basically characterized all these chemical compounds, and he began to focus on actually the field 4 or 5 because we wanted to publish papers. Right? We can't just play around with the compound forever. So this is a temperature compound we screened out. And typically, the way you want to identify target is to use something called target identification, right? So you attach biotinylated group to the compound, right? But before you do that, you have to make sure you're not disturbing the active group, right? So to do that, you can find several chemical compounds that share very similar structures. So this is the active compound. But if you have a chemical group right here in the middle, the compound is no longer active. That tells you that this part is very important. Don't mess with it. Don't mess with it, right? And then if the group is on the side, the compound is still active. So you know that this is less important. And whatever you do, you can do it here. You can attach the biotinylated group right here, right? So he did that actually. Well, he didn't do that, but it's done by a collaborator, the chemist J.J. Chen, whom I'll acknowledge in a second. And then by using this, we can do a put-on assay. And we actually identify several proteins. And he knocked them down one by one and identify a target A. I can't tell you the name because it's on the patent application. So I'm not allowed to tell you what the molecules are. Unfortunately, I did that. But we're able to really identify the target of the chemical compound. And this is something you would not suspect and something entirely novel. And then by doing this, we're actually working on 4 or 5 chemical compounds. A lot of work to do the chemical synthesis. But we're really able, by using this sort of a large scale screening, number one, we're able to identify more chemical compounds that could alter the fate of ES cells. So we envision that people can use this compound in the future to manipulate cell fate, and we cannot handle all these compounds. So it's going to be public information very soon. So, and number two, by focusing on a few compounds, we can identify or we can achieve new information about how this signaling network works. As you can see here, we identify new molecules. We're going to go deeper to understand why these molecules are important in the future. And then, so this is basically my summary slides. So I think to understand how human ES cells or how human pluripotent cells work, we're going to have to take an integrated approach. And I think this is in keeping with the goal of EBIX and also this particular workshop. So you're an engineer, but so what? And you can still be part of this work. I collaborate with a lot of engineers on campus, and you see the name in the next slides. If you decided to work alone by yourself, I think it's bad news, and you cannot handle this challenge, right? So, for instance, if you study human ES cells, you can understand long-term cell renewal, and then you can understand direct differentiation. But what are you going to do here, right? Once you have differentiated cells, you're going to have to work with tissue engineer or even clinicians to figure out how you're going to use these differentiated cells. Right? We're actually going to hopefully get to that stage very soon. So for application, and then we envision that this information we're getting can also promote the reprogramming of somatic cells. I mean, as you could tell, whatever you study here is going to be important for the ES of identity, right? So it's understandable that some of the molecules or pathways might be conserved in the reprogramming pathway. And we actually showed that in a paper that being reviewed, that E-coherent is actually important for reprogramming. Unfortunately, there are 3 other people who actually showed the same thing. So we'll see. And what I mostly focus on are chemical factors. I didn't even talk about mechanical factors. And we actually have ongoing collaborations with Dr. Wang here at UIUC to understand how substrate stiffness can influence sulfate. They have a very nice story, ongoing story, to show that if you manipulate sulfate, manipulate the stiffness of substrate to a softer one that resembles the in vivo condition, you can actually protect the cells much better, protect their potency much better. It's fabulous work. So, like Raj was asking, so you can really study how this chemical and mechanical factors work together, integrate together to maintain potency. So again, the bottom line here is that by doing basic science, you can really use this information to manipulate, to understand and manipulate sulfate. And more importantly, once you have something you like, you have to work with other people, with engineers, with clinicians. So if you're an engineer, well, please don't feel you're excluded. I mean, the cell biology part is being worked on extensively, but everybody has a role in this very important process. I think without the involvement of engineers, we're not going to make a very rapid progress or huge progress. So I wanted to finish my talk by thanking people in my lab. This is basically, well, I didn't want to thank myself actually. I don't know why my name is actually here. So, sorry. So I guess this is sort of a copy paste from my lab. And Jesse is a very talented postdoc in my lab. He's actually going to go for interview back to China for interview on Thursday, and he has 7 interviews. And he's single-handedly responsible for the PNAS paper and also the compound story. And I didn't have a chance to present Dr. Dominic's work on the coherent aspect. AJ Gung is the graduate student who single-handedly worked on the screening project. Amazing student. Very fortunate to have people like these guys in my lab. I also wanted to thank my collaborators. You can see many people from UIUC, Dr. Jie Chen, and Dr. Tetsuya Tanaka, and also Dr. Genbin Chen, who help us with chemical synthesis. And then a lot of other people. Actually, this is a very incomplete list because I have so many collaborators now. And then I'd like to thank my funding agencies, NIH, and also the state of Illinois for giving me some money in the very beginning, but the state is broke, unfortunately, so they don't have money to give. I'm very sad, sorry. So the money is great, it really helped me a lot. And National Science Foundation, and very importantly, the Science and Technology Center. We don't have the money yet, but hopefully we'll get it. Anyway, and also the National Science Foundation of China, and Beckman Award from UIUC. So thank you for your attention. Yes, please.
If I understand you correctly, those stem cells can be regulate correct?
Can what?
Do those stem cells tend to aggregate?
Right. So the question is, do these cells tend to aggregate together? The answer is yes and no. So depends on the condition. So if you grow them on coherent conditions, if you grow them on dish, they okay. Let me back up a little bit. And there's a very important point I wanted to make. So the cells have to be, under most conditions, grow as clusters. I think that's why you asked that question. So it has been shown that cell-cell adhesion or cell-cell aggregation is a very important survival factor for these cells. In other words, if you grow these cells as single cells to begin with, a lot of cells will die. Actually, you only have probably roughly 10% cells that remain to survive after 24 hours. Many other single cells will die. So this is called clonal dependence of human ES cells. Well-known fact. Thank you for asking that question and allow me to make that point. So, however, there's a ways to increase survival rate, and there's a very nice Nature Biotech paper published 4 or 5 years ago to show that a ROCK inhibitor called Y compound is able to increase the survival, single cell survival, from roughly 5% to 30%. So you can do that. However, under normal conditions, we're still grow the cells as clusters. In other words, when you subculture the cells, you don't tritonize them to single cells. You cut them into clusters. You scrape them into clusters. So when you play them cells in the beginning, they contain roughly 30, 40, or 50 cells. So that way you have a much higher survival. So coming back to the other question, so do these cells tend to aggregate? Yes, if you grow them as aggregates, they're going to maintain as aggregates. However, if you, going back to the EB condition, so the way EB aggregates is basically that you don't allow them to grow on a adherent condition to begin with. They aggregate in suspension. So I think without making the story too complicated, I'll stop there. Okay, so they're cultured as clusters and they grow as clusters. And then they can aggregate in suspension when you grow them on low adhesion conditions. Okay. Yes?
Since you have mentioned there are some
Sure.
such as some substrates, have you ever considered that the cell counting force may influence cell?
Absolutely. Cell adhesion is essential. Going back to the adhesion story we have, if you block cell adhesion, the cell would downregulate OX4 and SOX2 and NALA, indicating that cell adhesion is essential for the stability of this transcription circuit. So we show that in that paper that is being reviewed. The mechanical property. So again, let me back up a little bit. So as a cell biologist, or as someone who wants to be a cell biologist, when you talk about cells, microenvironment is very important. So what are the factors in microenvironment? Soluble factors. Right? Imagine your cells in a dish or in somewhere. Soluble factors, cell adhesion, cell matrix adhesion are the three major factors. Mechanical factors are also important because they're part of the cell adhesion and also cell ECM adhesion, right? The stiffness and stuff are also. So if you think along that line, yes, mechanical factors are very important. So I think you mentioned the cell adhesion part already. But matrix stiffness is a crucial regulator. So I'll take 1 minute also to quickly tell you why this is important. So there's a line of high-profile papers published in the last 5 years to show that if you change the stiffness of substrate, you can either promote differentiation or protect pluripotency, depends on the cells, right? And there's a cell paper and all kinds of nice papers. And what Dr. Wang has shown on this campus is that if you apply a cyclic stress to mouse embryonic stem cells, cells will differentiate. So they're very sensitive to this mechanical factors, essentially. In a paper that is being reviewed by Nature Methods, again by Ning's group, they show that if you reduce the stiffness of substrate to basically to their in vivo degree, they're very stable. The output expression is very high, higher than basically the normal condition when they grow on hard dish, right? And then furthermore, you can bypass the requirement of growth factors. This is so important. So normally you need some growth factors to maintain pluripotency of mouse ES cells. Now without that soluble factors, this stiffness changes would be sufficient to maintain the mean pluripotent condition. This is the Peckley work, essentially. So you could imagine that all these factors are going to be important, but under a certain environment, some factors are probably going to play a more dominant role than the others, right? So some will probably dominate the other, depends on the microenvironment. Again, I think the integration will be important, right? So if the signals are weak and they have to work together, but the signal is really strong, they can dominate and sufficient to maintain a certain phenotype. I think the integration between mechanical and soluble factors are going to be a very important area for research in the future. Right?
Regarding that, is there any chance that the myosin inhibition has something to do with the ability of the cell to sense the mechanical stiffness of its environment?
Right. So what we know is basically that if you take myosin away, actually myosin 2A, not 2B, cells would lose OX4 and SOX2. So what we do now know, I mean, we know that you can hear the downstream factor or the modified target, in a way that if you maintain you can hear, going back to the signaling question, right? You maintain you can hear activity, you take myosin 2 away entirely, cells are okay. So this is basically meaning that what myosin 2 normally does is to maintain you can hear, right? So if you can do that artificially, you can protect the cells from differentiation. But we do not know what regulates the myosin 2 activity. Or what maintain, I mean, I don't think anyone knows that. I mean, a lot of people know that myosin is important, regulating mechanical properties of cells, but nobody knows how myosin activity is maintained. I think, or regulated, or local activity, or spatial activity, or temporal activity are maintained. I think it's a difficult question, but I think that will get to the core of the question. Yes.
It's a very interesting talk. My question is very general. In your work, you try to using the chemical factors or small molecule compound in the culture medium to regulate the cell behavior and the differentiation and proliferation. Another approach is a deliver like a gene. So can you comment on
Absolutely.
those two approaches?
Okay, so this is something people have been looking for many years. Study for many years. Yes, you can absolutely manipulate at the gene level to alter cell fate. There's no doubt about it. The most dramatic example is the formation of iPS cells, right? So that's an outstanding example of how successful that approach can be, right? So in the pluripotent stem cell, in human ES cells, people have been looking for pathways that are important either for pluripotency or anti-differentiation. For instance, if a gene is important for absolutely for differentiation, if you take that gene away by knocking down, for instance, your approach, can you maintain this? Can you enhance undifferentiation? Number one, so make culturing these cells easier, right? Because the cells spontaneously differentiate all the time. We make it look like the cells are easy to grow. They're not. I mean, they can differentiate on any given day. So that's why I think Ying's discovery will be very useful in the future. If it can be reproduced in human ES cells, that's what we're doing now as we speak. Yes, absolutely. If you can manipulate at the gene level by either knocking down something or get rid of it, knocking down, knocking out, or use chemical manipulation to that gene, it will be tremendously important. People have tried the Wang signaling pathway. Unfortunately, that became a little messy. It's not clear. The role of Wang signaling pathway in ES cells is not very clear, but people have been really trying very hard to manipulate that signaling pathway, right, beta-catenin and stuff. So I think absolutely that overexpression or knockdown can be something that can be quite important for manipulating cell fate. My only worry is that now people have been trying to get away from genetic manipulation simply because people hate the idea of manipulating genome, right? I mean, you overexpress a lot of changes, right? That's why in the IPSL field, people introduce genes and take them out so that you don't introduce secondary defect, right? Nobody likes the idea that you have 4 genes in your genome, right? That can be turned on at any single time. So that's something I'm really worried about. But you can knock out, for instance, very cleanly, and then if it can really promote some fate decision that cells are able to make over them. So yeah, absolutely.
So can we say that the chemical factor approach is a safer because the cell uptake from the medium?
Right. That's what people argue basically. Right. That's what people argue.
How about the efficiency? Which way is better?
Right. Chemical compound is very. I mean, they're very self-permeable. I mean, in general, I think you can achieve 100%, right? Depending on chemical. But small molecules, they're very self-permeable. Genetic manipulation remains to be challenging in ES cells. Actually, that will be something I think people like you or other people can work on. If you are able to achieve the efficiency of more than 50%, it will be really, really nice. Actually, the best gene delivery methods for the ES cell field will be fusion HD. Actually, fusion HD is actually 20% only. A max nuclear factor is 20% ballpark too. So we use lentiviral infection because we can infect with high titer. But for transient gene delivery, I just don't think there's a very good way to actually do that.
Thank you.
Sorry, I'm an engineer, so engineers.
You're the smarter guy.
Engineers like to design things, but also manipulate things. Suppose you have the cluster of cells. You want to target a subset of these cells and change them and decide the fate of those cells. Can you do that?
Yes.
If you do that, what do you expect to see for the cluster?
You expect this sort of question from a smarter person, right? So this is very philosophical. Again, going back to James' question, I think you can manipulate at a gene level and also a level of chemical. I mean, which is microfluidic device to deliver, at a certain precision and stuff like that. And then I envision. I mean, I don't have it, but for instance, for clusters, you can introduce an inducible system to certain cells, right? And you mix them together, but then you can apply some chemical compound that known to trigger this expression, right? And then so that the end results is you only have the expression in certain cells. But this is under the premises that you are able to deliver that inducible system in certain cells to begin with. Right? So I think Law B is something that can be quite useful. Right? You mix them together, you do some manipulation so that certain cells have certain genes that can be turned on, right, by something. And being part of the STC myself, I've learned a lot about microfluidic device and stuff. I think that will be something very, very cool. Chemical scaffold and microfluidic device allow you to deliver in a micro scale, like what James was talking about, to in a very targeted way, to again apply shRNA or genes for overexpression or chemical compound that can be localized very, very precisely to trigger some pathway. I think that can be totally done in typical.
Another question I have is, I was a little surprised that individual cells, these are precursor cells to ES cells, right? Or they are ES cells.
The cells we begin with are the bona fide ES cells. They're just ES cells. But the new progenitor cells we use this particular chemical compound. They're newer progenitor cells. They're somewhere in the middle. So ES cells, from the development point of view, this is embryo. This is precursor and this is adult, right? So we're able to do this in a very high efficient fashion from here to here. But here's a mess. That's why I think this STC can be very, very important. That's why I'm so excited to be associated with the STC, because if we're able to, for instance, promote with high efficiency the formation of DA neuron or motor neuron. Motor neuron will be challenging because survival is a huge problem for everyone. DA neurons with 80% a chemical compound or something, I think we'll become millionaire.
Okay, but I was a little surprised that you mentioned that these cells, individual cells, only 5% of them will survive, unless you have clusters of them.
Yes.
If that's the case, we know all embryonic stem cells, they started with one egg, right?
Right.
One single cell. And if only 5% of them survive, then the infertility rate must be very high.
I agree with that, but only under in vitro conditions. So somehow we're not able to get the condition right. I think Stephen will be so excited. I mean, I can't wait to hear the results from my students. I think whether it has effect on survival or anti-differentiation will be amazing for me. I mean, I just think there's some pathway we're not able to recap entirely, right? There's some factors there that promote survival. We're not able to identify, right? That's why I think it's.
Does it have something to do with the first trimester? The first 3 months is the most important time.
Now you become a reproductive biologist.
Beyond that, the embryo is pretty stable. But before that,
I wouldn't translate that 5% in vitro sort of rate to. I would be hesitant to do that actually, because I think if we say that, it's going to be too scary for a lot of people. I just don't think it's true actually. I think it's just a matter of fact that in cultural conditions are not capable of reproducing what happens in vivo. But I think you made a very good point that there's something there about the fragility or whatever. These cells being fragile somehow in the very beginning. I have no idea how that works.
So if you have a cluster of ES cells and you introduce one or a bunch of cells that are further along in differentiation, do those more differentiated cells somehow induce any kind of symptom?
Okay, so are you referring to potential heterogeneity or I probably didn't get the core of your question. So
no, I'm just asking if anyone has tried to have, say, a cluster of ES cells,
okay,
and then introduced in that cluster a single cell or a bunch of cells that are more differentiated. Does that affect the initial ES cells?
Very good question. So the question, if I can rephrase, is can you influence the other cells by introducing the cells that are a little different at the stage of development or something different? Can you influence other cells by introducing a cell that is different? So absolutely. I think the potential is, I mean, we're talking about paracrine or autocrine loop. Yes, absolutely. But it depends on the feature of that cell. can the cells make something that is important, right? Yes, on paper, absolutely, yes. I mean, if that cell can make growth factors that are important for something, right? I mean, that would be really, really nice. Yeah, I agree. I mean, I think a simple version or applicable version of your question is, if you have two group of cells, can you take the supernatant of that cells and apply it to the other cells and see whether you have influence? I think that would be a good experiment, actually. I mean, yes, absolutely. I think that's a good experiment.
And maybe the waste generated by one type of cells, food, would be food for the other cells.
I'm thinking about your progenitor cell condition medium and put them in ES as I'm just answering this question. That's what I'm thinking, actually. But never mind. Yes.
So when you make these neuroprogenitor cells, are they still proliferating? Do they still
Well, good
multiply
question.
more?
We haven't. So the question is, do these cells still growing more? I mean, they're differentiated at least halfway. Well, at the early stage, we really haven't done a lot of characterization. So we didn't do BRDU, which will easily address the question of PRX67. And my feeling is, yes, they're growing. They're still growing. They're still far away from maturation. From there, it will take at least 2 weeks for them to become mature neurons. They're still lacking the expression of certain markers without getting into the details. So they're your progenitor cells.
But are they becoming neurons on their own, or are you inducing?
You have to, again, in an in vitro condition, you have to manipulate. Otherwise, I don't know. I mean, I haven't really allowed them to grow for a little bit longer time. But I know that Steve Stice, another person in STC, generates stable cell lines. So I think, you could potentially grow them forever, I mean, for 30 passes.
So you haven't tried any cold cultures either with other neurons or astrocytes?
This is, I'm waiting for the money from you.
Yeah, this could be a very naive question. Do you know why the human embryonic cell is so difficult to handle? I mean, they have to stay with the culture, but compared to the mouse embryonic cell, even the primary cell, the cancer cell, they are much more robust.
So so I think I'll try my best to address that. Okay, number one, they're still primary cells, so to speak, right? I mean, they're very special primary cells because they can grow for a long, long time, right? So that's the point I was trying to make in the very beginning. So primary cells are difficult to transfect to begin with, but there indeed something more special about these cells, in a way that there's might be permeability issue. That's the first thing I suspect, actually, because we know certain compound that we applied didn't really work well. I didn't really know whether that's because the compound doesn't work or actually doesn't even get to the cells. One example is warp-manning. I'm never able to get warp-manning to see some AKT. I mean, so IOI works really well. So I have no idea. So there's something about maybe the membrane of the cells or something. There's some
Not for I'm talking about the cell itself. You take out from you mentioned the feeder cells or the matrix cells. They don't last very long
well,
compared to other type of cells.
like I mentioned before, nowadays you can grow these cells even without feeder, so that's not a problem. So you can grow them using feeder condition medium, right? So you basically can still have the single cell, so that's okay. That doesn't bother us. I mean, you can even grow these cells in chemically defined medium and feeder medium. So, but whenever you do transfection, there's always problems actually. So there's something intrinsic about the cells, other than that, they're primary cells. Thank you.